You're recording now.
Great, thank you. All right, so in today's lecture, we're going to do something that's a little bit outside the normal scope of the course, and we're going to talk about some very recent developments, understanding predicting protein structure. And some of you may have seen some of these announcements that came out both in the scientific press, such as these articles in Nature and Science, and also in the popular media. And a lot of them are quite hyperbolic. 50-year-old protein folding problem is finally solved. That sounds mild compared to what Nature says. It will change everything. The game has changed. And what I wanted to do today was take you through some of the background of this, the process by which programs for predicting protein structure have been evaluated over the last almost 2 decades now, and to really show you how this is a rather dramatic advance, and to tell you what we do know so far about what's been accomplished. And I should say that a lot of the information is still not available. So we'll tell you what we do know, and hopefully more will emerge in the near future. We'll start with discussing these things called the CASP challenges, which is the challenge through which it emerged that this program is so much better at predicting protein structure. And this was an idea that was set up some time ago and really has become key to the advancements in protein folding. And we'll look at what some of the long-term winners have been in this, and then we'll look at Google's contributions. They made an important contribution 2 years ago, and now for which we have some more information. And now there's the one that was just announced in a press release earlier this week. So CASP is shorthand for Critical Assessment of Techniques for Protein Structure Prediction. And it was an idea that came about in the 1990s. There were a lot of people who had been developing programs to predict protein structure, and it was impossible to tell whose programs are doing better than anybody else's. And the rather clever idea here was to get experimentalists who were in the process of determining protein structures to let people know what structures they were planning on solving over the course of a year or two. They were already working on. And then the CASP organizers released the sequences of those and asked the scientific community to try to predict them. And so no one but the experimental group would know what the right answer was until the end of the contest. And even for most of that time, the experimental group didn't have the answer because they were still in the process of solving it. So this gave a very rigorous way to evaluate whether a program was actually doing a good job. And then they would score the different structures based on how difficult they were. And they developed very rigorous methods for evaluating by many different criteria. And so this, every 2 years since the mid-1990s, they've been running these CASP challenges. And for quite a long time, the single best approach was the one based on Rosetta, which is something that you've worked on with quite a bit during this course. And I'll take you through the way Rosetta described their programs for predicting protein structure in a paper about 2009, which was when they had already emerged as one of the leaders of this field. So in a lot of cases, they can take advantage of homology in predicting protein structure. And the idea is that in the challenge, you're given a sequence. They did sequence alignments and found homologs of known structure. And then they would, when there was a good homolog, they could mutate that structure and refine it using techniques very similar to the ones that you used in your projects. Now, this depends on there being good homologs. And this pie chart from that paper shows that at the time, at least, there were about 16% of the proteins that were really, really tough. There was less than 20% sequence homology. But for roughly half or more of all the proteins, there was some reasonably good structure from which you could start. So that was really great. That's for human sequences. And if you're interested in sequences from other species, the situation can be quite different. So on the top here, you see a pie chart for the malaria parasite. And in this case, almost a third of the sequences are hard, and only 2% are easy versus 16 hard and 14% easy in human. So what happens when you don't have, I should say first, so the refinement procedure then, when they do have a good structure to start with, they do random changes to the backbone torsion angles. They do rotor optimization on the side chains. Again, all of this should sound very familiar from the work that you did at the beginning of the semester. And then they did energy minimization on the torsion angles. And depending on how close the starting structure was in sequence homology to their candidate, they had slightly different versions. And so they had a version of this refinement procedure when there was over 50% sequence similarity, another one for 20 to 50, and then yet another one for the low sequence similarity. So this should make a lot of sense in terms of the things that we've already seen in the course in terms of the ability to take a rough structure and refine it. Now, what happens when you don't have a good starting sequence? So here they had another really clever idea that became quite influential. And when there was no homology model, they would do a Monte Carlo search for the right backbone angles. And they didn't start that with a random search, but they would take short regions of the sequence and look in the protein structure database for any other protein that had the same short little sequence, 3 to 9 amino acids. And they would make a library of those little segments. And then they would set the torsion angles of the target protein to the ones from the similar peptide in the PDB. And they would use the Metropolis criteria that we've talked about to optimize over that. And you needed to do a lot of Monte Carlo steps, right? So it's 36,000 steps. And you can imagine that this is quite a large search space to go over, but nevertheless, it actually produced remarkably good results. And they didn't just do that once, but they did it 2,000 times in different starting structures. And so now you get a family of solutions. And with that family of solutions, you can see how similar they are to each other, so how consistent the result is. And you can get a sense of what parts of the structure are better determined by this process than others. And finally, they would cluster those, and so there might be more than one solution that would be discovered. And in the last phase was refinement with the same kind of methods that we've seen that use potential energy functions to do the fine-tuning of the structure. This is a plot. I'll show various versions over the course of the lecture, in which they're using in this CASP evaluation a criterion that they call GDT_TS. And you can understand that roughly as the percent of the residues that are correctly modeled, where they've taken out the things that are really easy. So 100% accuracy is at the top here, obviously. That means you've gotten every single residue right. And in these plots, they typically show the GDT_TS as a function of the target difficulty. And they've got well-defined metrics, which I'm not going to go into, for figuring out how hard a target is. And then what you typically see over the different years of the competition is that it's a lot easier to solve some targets than others. And this target difficulty, there's usually a fairly good linear correlation. So in the first CASP challenge, what they called easy targets, they were getting right 72% of the time. And some of the tough targets, they were getting right roughly 26% of the time. And as the contest proceeded, you can see that they're making significant improvements in these hard targets. So over the first 10 years, they went from getting 26% of the hard targets right to getting 49% of the residues in the hard targets right. Now, that was the first decade. They went from 26 in these hard targets to 49%. And then the next decade, they only made a really modest improvement, right? So the roughly 49% of the residues were right in CASP 6. And by CASP 10, a decade later, 55% of the residues in these hard targets right. And there was really a feeling in the publications that you see from this that the field had stalled and that something really had to be done to shake things up. So this is a more detailed version of that same plot, where each protein that was a target that was solved is indicated here by one of these marks. So the triangles are from CASP 10, and the circles are from CASP 11. The purple line is the regression for CASP 10. The black line is the regression for CASP 11. They really don't look that different, right? So over the course of those 2 years, really nothing had changed there. But there were some things that were improving. And they point out that they're able to do much longer proteins in CASP 11 than they were in CASP 10. So that was a good sign. And there were a lot of technical things that seemed to be improving a lot. And the one that's going to become really important as we think about how Google is successful is this idea of contact prediction. So contact prediction was improving. We'll get to exactly what that is. And then there were methods for refinement that were improving and how to handle places that were not in the template. But that backbone accuracy, which is what was measured in that GDT_TS versus a difficulty plot really had stalled. So contact prediction really is one of a really early idea in predicting protein structure. And one of the early papers is this paper from 1994. And the idea is a very intuitive one. So if I have a protein of unknown structure, I look in the sequence databases and find all the homologs. And so this block down below is a cartoon of a multiple sequence alignment where the protein that we want to solve are the light blue squares, and then lots of the homologs are lined up beneath it. And if you see two positions in this multiple sequence alignment where there seem to be correlated mutations, that might suggest that these two residues are interacting. So anyone remember there are single amino acid codes? Someone want to volunteer what they see in this pattern of sequence alignment that's shown at the top?
K is lysine and E is glutamine, something like
Good,
that.
right. So what's the property there that you're seeing?
So K is positively charged and E is a polar residue.
Oh, yeah,
Or
so
maybe it's also negatively charged.
it's negatively charged, right? So that's a complementary charge pair. Anyone remember what L and A are?
Leucine and alanine.
Leucine and alanine. So what's the complementarity there?
Seems like both of them are nonpolar.
Perfect, right? And so you might see patterns like this where when one residue is nonpolar, the other one is nonpolar, when one is charged, the other one has a complementary charge. And that could suggest, if you see this over the entire alignment with many, many sequences, that these two residues are interacting with each other. So that's the core idea. And there are a lot of variants on this. And that early paper in '94 had one version. And then there was a decade or so of research on this. And this shows the precision of these predictions using various algorithms from the CASP challenges as a function of the depth of the sequence alignment. Okay? And the dashed red line is the regression line for CASP11, which already was an improvement over previous ones. And then you see CASP12, they're making even bigger improvements. Right? So when you get to really, really large sequence alignments, which are the far right-hand side of this plot, then it's pretty easy to find residues that are contacting each other. And the challenge is when you have very low depth in your sequence alignment, very shallow sequence alignments, then it's harder. And so this really became a very important piece in a lot of prediction tools. People realized that they could leverage this. And what's the advantage of using sequence alignments? Well, with the sequencing being cheap, the number of sequences that we have in the databases keeps growing at a very, very rapid pace. And so increasingly it becomes that we're working in a regime on the right-hand side of this plot versus the left-hand side. So what they say is that in the summary of the CASP challenges that was published in 2017, that most groups had come up with new methods. They were doing really well on some targets when the sequence alignment is really deep because of their ability to predict these contacts. And they were impressed because there's an example, this was the clearest example, they say, where a theoretical advance led to improvement in model accuracy. So you develop a better way to use multiple sequence alignments, and you get much, much better structures out the other end. Any questions on this so far? Okay. So here again is one of those plots of GDTTS, the backbone accuracy. Here it's plotted against domain length. And another thing that they were impressed by is their ability to start of the field to start dealing with sequences in the far right of this plot, which is much longer proteins. So you can see in CASP10, the longest proteins that they were solving structures for were about 200. And by CASP12, they're solving structures that are 350 amino acids long. And now the accuracy is dropping, of course, the longer the protein is, but still, even at 250, there are a number of structures that are being solved with pretty good accuracy. So this, at the time, was a cause for some optimism compared to CASP11, where it really looked like things had stalled. CASP12, there seemed to be some modest improvement. CASP13, though, is where things start to really take off, and you can see it from these regression plots. So 11, 12, they're all down in the same pack where it looks like you're doing, pretty much the same as you always had in terms of the relationship between accuracy on the y-axis and difficulty on the x-axis. But in CASP13, you see this plateau where the best algorithms are able to predict structures of really hard proteins with something like 60, 70% accuracy. And a lot of this is attributable to the ability to get the context accurate. And so here's the plot for the accuracy of contexts in CASP9, 10, 11. You can see it always, was below 30%. CASP12 seemed like a big boost we had talked about, but now they're getting roughly 70% of all the contacts correct. So that was one important aspect of this. But the other really important aspect came about last contest, 2018, with the entry of Google into this. And there were a lot of hyperbolic headlines of the type that we've seen in this current round as well, about Google's DeepMind acing protein folding and how it's going to give pharma a run for its money, and how they've cracked a problem that has long vexed biologists. And you can see how much better they were doing from others in this plot here. So this y-axis is the number of domains that don't have templates, that don't have good homologs with known structure. And the x-axis is how accurate the structures are. So you can see that, for example, if we just follow, say, the blue line, they get more than 40 domains they get with an accuracy of 20 on this scale, and at an accuracy of, say, 50 on this scale, they're getting about 30, right? And very few are greater than 90. But nonetheless, they're far outstripping the rest of the field, right? So it doesn't take a lot of detailed analysis to tell that this group is really doing much, much better than all the others. So what do they do? Well, when they, excuse me. When they published the results from CASP, from the CASP contest in 2018, we didn't really have a lot of information to go on. There was a blog post, then a paper came out in 2019. That didn't really match the blog post or the abstract. It was all very, very confusing. In describing to you what they did, I'm going to focus on this paper that came out earlier this year, which describes what, at the time, they thought was the best approach they could do that was consistent with what they had done in the contest. But during the course of the contest, they had changed their approach over time. And that's kind of the nature of one of these longitudinal contests, that you get ideas along the way, and you keep implementing newer and newer ideas. So we won't try to figure out everything that they actually did in the contest, but just focus on what they described as the best thing as of, I think it was January of 2020. Core idea here is really still a very simple one. They start with this idea that we already saw about using multiple sequence alignments, MSAs, to get information about what the protein structure might be, in the same way that that paper from 1994 had tried to predict contacts from a multiple sequence alignment. But the difference here is that they feed it into a deep neural net, and I'll explain a little bit what the neural nets are. And that neural net takes the multiple sequence alignment and gives them predictions of the probability that two residues not going to be contacting each other, but a probability of the distance distribution over those contacts, and a probability distribution over the torsion angles of those contacts, of those residues. And then they do a refinement using a new potential energy function. That's not the potential energy function that we saw early in the course from, say, Rosetta or CHARM or one of those, but it's a potential energy function that derives from this deep neural net. Is this overall scheme here clear? Any questions? Okay, so deep neural nets, I'll give you the 2-minute version of what a deep neural net is. Obviously, there are whole courses you can take on this, but the fundamental thing to realize about a deep neural net is that it's a tool for discovering some unknown function. So if I have a training data set and some output, so in our case, what's a training data set? It would be that multiple sequence alignment. And what's the output? It's a structure. And I want to find a function, obviously it's going to be a very complicated function, that takes as input the sequence and gives me the PDB coordinates as the output, right? That would be magical if you could do it, but it's in fact what they're able to accomplish. And neural nets are capable of finding very complicated functions that can be highly nonlinear. The key is that you have to provide it with these training data. So you give it an input and output, and it learns the function along the way. And then you can evaluate how well it's doing, not by how well it reproduces the data that you've already given it, that you've trained it on, but a different data set, a test data set that you've held out. So this is often done in a cross-validation approach, or you can have a permanently held out test set if you've got enough data to do so. And then through this process, you can determine with some accuracy whether this algorithm will generalize to another data set or not. So we've seen things like this throughout. You've done a lot of fitting of curves, differential equations, linear models throughout. The goal is exactly the same as any of the fitting that you've already done in Python or MATLAB. The key is that here we don't have to tell your algorithm what the form of the equation is. We don't tell it it's a linear function. We don't give it any information at all. It discovers the right kinds of functions on its own. And the neural net is made up of many different functions that are used one as input to the next. And it's called a network because it's easiest to describe what these functions are in a graphical representation. So if you look at this graph on the bottom, you've got some input and that goes into the first function. The output of the first function goes into the second function, and so on and so forth. Now you can think of the input here as being one of thousands of variables, and for each one of those variables there's a path of what function goes into next, and then what happens after that, and what happens after that, and can get rather complicated. So writing out a set of equations for all of that becomes primitive, but you can look at it graphically in terms of which function is feeding into the next as a network. These intermediate steps are often called hidden layers because you're given as the training data, the input and the output. You don't know any of these intermediate variables, which are going to be discovered by the algorithm finding out what these functions should be, f1, f2, f3, and then computing what these intermediate values would be. How does it do it? Well, there's a loss function that is the difference between the algorithm's output and the real output. And it's actually a fairly complicated set of algorithms for optimization that try to adjust all of the parameters and the structure of this network to minimize some error between the input, between the true output and the predicted output. It's called neural networks for a fairly arbitrary reason. There is a rather weak analogy between how each one of these units in this graph functions in terms of how it weights its inputs and output to produce an output, and how people believe neurons fire. It's not a particularly useful analogy in most cases when you're actually trying to make sense of a neural network, but that is the origin of the field. And I guess the final piece to realize is these neural networks can be very, very large in terms of the number of hidden variables. And so there'll be layers of functions. Each one of these layers represents a set of functions, and they project from one layer to another. Means this function takes as an input only some of the functions from the previous layer, and so on and so forth. This function over here takes as input only some of the things previously. All right, I realize that was a super fast explanation of neural nets. Any questions? All right, so let's see how this actually plays out in this AlphaFold algorithm. So the key thing that they did was they decided to try to use neural nets to produce a potential energy function that would take a sequence of a particular protein as input and give you the potential for different structures. And they parameterized that structure as a prediction of protein backbone angles, and the residue distances. So we talked previously about the residue distances as the contacts, and here they're also going to produce a potential energy function over the backbone angles. And the truly amazing thing that made this such an advance was that when you correctly specify this potential energy function for your specific protein, you can carry out gradient descent on that potential energy function and go from an unfolded protein to the right answer. And just to, let it sink in, like how big a deal this is. Remember when we had our generic potential energy functions that were trying to capture all proteins, and we wanted to say optimize, phenylalanine that had been flipped 180 degrees, you couldn't minimize that, right? Because it would have to overcome some energetic barrier to get from one angle to the other angle. But here, this potential energy function is smooth. So you can get from the wrong torsion angle for a side chain to the right torsion angle, but you can get from a completely unfolded protein to a folded protein just by following the gradient. Now it seems too good to be true, but we'll see that it's actually how it works. So this is the structure of that neural network. The input is on the left-hand side, the output is the right-hand side. The input are the sequence of the protein that you want to predict and the multiple sequence alignment. And the output is going to be, for every pair of residues, what's the probability that they're at a specific distance? And what's the probability distribution over the angles, the torsion angles? They analyze the multiple sequence alignments in windows that are 64 amino acids long by 64 amino acids long. That's what this dashed box is supposed to represent. So you're sliding this window across the multiple sequence alignment. And then, as I said, they're not going to predict which residues contact each other in a binary way, but they're going to predict a probability distribution that the distance between residue I and residue J is whatever it is. So it's a continuous probability distribution given the sequence that you start off with and the multiple sequence alignment. So that's the output. And the other aspect of the output is the fine psi angles, so the backbone angles. Again, it's a probability distribution over fine psi angles, not a fixed prediction. So here's an example of the results. And this is the actual contact distances for a particular protein that was in this, I guess, is CASP13 challenge. And here they're showing the mode of that probability distribution, right? So they're not, remember, they don't predict precise distance, but they predict a probability distribution. The most probable distance for any particular residue is shown here. And it does, at first glance, they look almost identical. It's hard to tell which one's the actual distance and which one is the prediction. So this is really a stunning result. Remember, we took as our input the multiple sequence alignment, fed it through this neural network, and out pops a very, very accurate representation of the distance. Now, they're not the first to try to do something like this, but one of the key insights was not to just try to predict the distance, but to predict the probability distribution, because when you have a probability distribution, then you can take a derivative over that, you can develop a gradient. And so this plot shows probability distributions, in this case, for 41 different amino acids, their distance to or 40 amino acids, their distance to residue 29. So we just look at one of these boxes. On the x-axis is the distance. Looks like from 0 to about maybe 20 angstroms. And the y-axis is the probability distribution. And so the red lines indicate the mode of that probability distribution. And what do you notice about these probability distributions? They're not uniform, right? So there's quite a lot of difference. There are some residues, like these down here, where there's a very, very strong peak. They can predict with high accuracy, with high specificity, whether they're accurate, we'll see or not. But they predict a very narrow probability distribution that residue 28 is going to be very close to residue 29. Well, that doesn't take a genius to figure out, right? But even for some of these amino acids that are further away from residue 29, you can see sharp peaks in some of the probability distributions, whereas in others, it's much more gentle. So say for residue 15, the algorithm is not very confident as to how far residue 15 should be from residue 29. Any questions? This plot on the left-hand side shows the mode, right, of that prediction against the true distance. And you can see it's really quite remarkable. They're getting extremely high accuracy. That's important. But the other aspect that may be almost as important is the fact that when the algorithm gives you a very, very sharp peak, then it tends to be right. And when it gives you a very broad peak, then its confidence, it's basically accurately measuring its confidence. Maybe that's the right way to say it. So when the error is large, the confidence is low. When the error is low, the confidence is high. And that's another property you'd like to see when you're trying to use that probability distribution to generate the potential energy function. Okay. So as we said, they're producing a distance potential. They also produce a backbone torsion angle potential. And that's what these two terms in the bottom of the potential energy function are, the V distance and the V torsion. But then they do add a third potential from Rosetta, and that helps them a bit because it avoids amino acids overlapping with each other, atoms overlapping with each other. So that's the van der Waals potential that we've seen before. So this is a more detailed schematic of what's actually going on here. On the left-hand side, we have the sequence, the multiple sequence alignment, and they use some fairly standard programs to do that. So there's nothing particularly creative there. The sequence database, that gives you the multiple sequence alignment features. And then they put that into a neural net. And another aspect of this, which is really very interesting, is they're using a fairly standard neural net. So ResNet is one that's extremely popular in image analysis. It's a fairly standard architecture, so it's not something that they crafted very finely. So the ResNet gets the individual sequence information that I got from the multiple sequence alignment, and it generates those two distributions that can be used to generate the potential from the torsion angles and the distance. And then, as we said, they added the Rosetta. That gives them the potential energy. Okay. And now, how do you actually get a structure out of that? Well, you choose some initial random conformation, and then you run gradient descent. Now remember, it's gradient descent in this special potential where only one term is a physical term, the van der Waals, and everything else comes from the neural net. And here's an example of one of those refinements where a specific starting structure is shown in the far left. And then you can see the refinement as it's doing gradient descent. And they've plotted in red the RMSD from the final structure. And then in blue is a measure of how accurate it is. So 1 is perfect accuracy to the true structure, and 0 is very bad structure. What do you notice about this in terms of thinking about gradient descent, right? Gradient descent, we always have to be uniformly declining, right? There's no way to go uphill in gradient descent. And so, interestingly, their gradient accurately helps you get over some things that are actually bumps, where the starting structure is a better match to the final structure than some of these intermediate structures. That's in this root mean square deviation space, right? In a three-dimensional space, if you're just trying to minimize the error in RMSD, you would not be able to do gradient descent, even if you had that function. But because of this complicated function that they've generated, they're able to jump over some of those intermediates. And so you can see that it descends rather quickly. And on the right-hand side, you can see the difference between the predicted structure and the accurate structure. And they're extremely, extremely similar. And this is within sometimes the error that you get when two different crystal structures are solved. So it's just a truly remarkable accomplishment. Another way to analyze this is by looking at what secondary structure elements are present at various times along the way. And so in red, they've got places where it's a beta strand. In blue, the helix. And this little plot on the right is the actual structure. And so what do you see when you look at, and then the left-hand, you're going from left to right, is the steps in the gradient descent. So what do you see in terms of when secondary structure forms and whether it's correct or not? Anyone want to try to describe what's seen here?
seems like they knew immediately where the alpha helices were, and then the beta strands were harder to predict.
Exactly. So you see that the alpha helices in blue, they get very, very quickly, they're right. They make very few errors in alpha helices. There are a few minor changes along the edges, right? Exactly where it starts and stops. And that's primarily just the definition of helices. The beta strands are harder. Sometimes they start off with something that's wrong. So this piece at the very top, so the C terminal end of the protein is predicted to be beta strand for quite a while before it disappears. Others, they start off, it took quite a while for these beta strands to form. So why do you think it is that it's doing better with alpha helices than it is with beta sheets?
Could it be that alpha helices are self-contained, whereas a beta sheet usually has to pair with something close by? So it's like 2 units versus 1.
Yeah, it was a little hard to hear, but I think you said alpha helices are self-contained and beta sheets have to pair with something further away, right? Yep, absolutely right. So where in their method is that local information? How does it relate to the method that they developed? And if we think back to what the method is, right, what are the key inputs here? It all goes back to the fact that the most of the information they're getting is from the multiple sequence alignment. And so they're being driven by contacts, and you'll have more close approaches in an alpha helix because of that self-contained nature, as you just said, than in the beta sheets, where there'll be fewer constraints to have the beta sheet be in the right place versus some wrong place. And so it's the contacts that are really driving this. And you can see that very clearly then from the fact that really easy to produce something correctly when you've got lots and lots of contacts. But if there are fewer contacts, it becomes much more challenging. So again, they're able to solve these kinds of structures purely by gradient descent because they have the right potential energy function. This would not work if you'd use the Rosetta potential energy function or the Explorer potential energy function. And we know that, firsthand from your own work, right? When you did fast relax, you don't get from an unfolded protein to a folded protein, no matter how long you run it. But because of the special potential energy function that they've generated, they really can go from these very, very wrong structures to something that's quite accurate. They're not happy with just one prediction. They start with many different initializations. And then through that series of initializations, you get a family of solutions. And then there's the final relaxation with our old friend Rosetta. So we haven't given up on the Rosetta system. So when you're done with that, what you end up with, just like we saw with the Rosetta that was winning in the 2009 or when it was, you get a family of solutions. But here, the accuracy of that family of solutions is so much higher. I think it's interesting to see how they describe what they did themselves. So here's a quote from I think this was their blog post, and they stress that they're building on methods that are commonly used, right? So there weren't any brilliant new insights here, but they used, and they weren't even the first to use neural nets, but they engineered it really well. And perhaps there were a couple of key choices that they made, but largely these were ideas that were already out there and they were just put together in an extremely useful way. So at the time that this result came out 2 years ago, there was a complicated set of emotions that you could see in people's responses to these things. Because remember, people have been working on trying to solve protein folding for 20 years or more, actually much more than that. Some of the early work was done by Cy Leventhal, who I got a chance to know when I was very young. And he had been working on these ideas since, I think, the 1950s. So people have been trying to figure out how to solve protein folding de novo for the better part of half a century. And suddenly this group comes out of nowhere with not a lot of expertise in protein folding, and they were beating everybody. And so there were a lot of reactions, and I think Al-Qureshi's blog posts were pretty useful in sort of articulating both the emotional response, but also putting the results into context. And he pointed out that some of the key differences between what they were doing and what other people were doing was that everybody else had assumed that the best way to do this was try to develop a universal potential for all proteins, and then use that universal potential to try to optimize your protein structure. And this approach said, no, let's just find out a specific potential energy function for this protein based on the multiple sequence alignment. So it obviously worked, but why was that a fairly radical idea? Why do you think it would be more natural to try to come up with a universal potential energy function rather than a protein-specific one?
Probably just the idea that all proteins follow the same laws, or you'd expect them to. So you would want a universal law to describe proteins if you could get it.
Absolutely. So one aspect is that, we expect they're all following the same rules. If we were doing this from a purely physics-based approach, we would assume that the physics is the same regardless of what protein we're working on. And so even though remember our dichotomy between the physical approaches and the statistical approaches, even though we're taking a statistical approach to the problem, we'd still expect that the same kind of statistical patterns would work. So that's an aesthetic reason, if you will, for wanting it. Are there any practical reasons why you think it might be easier? Obviously, it didn't turn out to be, but why you might have thought it easier to get a good statistical potential based on all proteins rather than individual protein?
Especially at the time that the competition started, there probably wasn't quite as much data. So it was kind of like nice to integrate all that data for one potential function.
I think that is a really key insight, that the idea that there's enough information in the multiple sequence alignment for a single protein, it seems absurd, right? It's just, it's not a lot of data. You would imagine that you have to do this over all the known proteins, and then you have enough training data to learn the function. And this is where the power of these neural networks really becomes clear, because there are some things that you can build off of that pick up signals. So the fact that they're using ResNet, which is a structure that's been used very successfully in image analysis for their neural net, the basis of their neural net, really gave them a boost. And if they were starting from scratch with no known architecture and trying to do it, they probably would not have been able to do protein-specific potential energy functions. So that's where things stood as far as the field knew until, I think it was Monday, when the press releases came out. And then these press releases started coming out. And as I said, in the popular media, it really became quite excited. So, protein folding problem finally solved by Google, or London AI lab claims breakthrough, a major scientific breakthrough. Right? It's seen as something that's a really amazing transition. And you can see that by going to these plots. So remember, this is the plot up until this current year. And you can see that, big advance in the first decade, slow advance over the next 10 years. Then CASP13, which is the one we just finished talking about, where Google made its first entry, seemed to be a big jump forward. So here's a less pretty version of the same plots. And this is CASP. So, all right, CASP13 is the red line. And so you can see that CASP13 broke away from all the previous ones. Then CASP14 is the green line. If Google had not participated, you would have seen, a significant improvement. But basically the trend line is kind of that everyone's building off of what was accomplished in the previous round. So CASP13 was the one that we just talked about with Google's first submission. They made this dramatic jump forward. And then, if they hadn't done anything new, the field would have made a modest advance. This is with their submission. You can see something dramatic is going on here. So remember, the x-axis is target difficulty, and the y-axis is the fraction of residues that you're getting right. And the truly astounding thing is, on the hardest targets, their accuracy is in this 85–90% range, which is what other algorithms are getting only on the very easy targets. It's just remarkable. It's so remarkable that it's really at first hard to believe that you can go from the level of accuracy that we were seeing before to this level of accuracy. So what do we know about it? Not a heck of a lot. So I'm going to take you through some of the things that have been published. Some of this is based on reporters talking with them. There's really not too much information on what exactly they did, but this is what I've gleaned from what's available in the literature. So there was a pretty good paper article in Nature that went online November 30th. They include this plot, which gives you sort of similar information to what we just saw. But this is that GDT underscore TS average score, so averaged over all the proteins. And you can see that for most of the CASP competitions, they're getting about 40 on this score. This was the Google solution the first time around, AlphaFold. The last time its average score was around 60. And now this latest submission, their average score, right? So that's averaged across all of these targets of all the different difficulties. Their average score is almost 90. And that's where you get to be almost as good as solving the protein structure experimentally. They, in this piece, say that nearly 2/3 of the structures were comparable to experimental structures. And Mould was the one who had originated, one of the originators of this contest. And they point out that it's not really clear whether that's because Google is wrong, or maybe the structures are wrong, because a lot of the structures that they're getting wrong are ones that are being solved by NMR rather than X-ray crystallography. And as you know from the very beginning of the course, we talked, Alan talked about how structures are solved by NMR and crystallography. They differ a lot in what they're doing. You have a lot more dense information in X-ray crystallography, so the solutions are much more likely to be accurate. With NMR, it is possible to have serious errors because you just don't have enough NMR constraints in some part of the protein. So it's conceivable that the real score for AlphaFold 2 is even higher than what they're getting here. It's not perfect. So where do they tend to mess up besides NMR structures? Well, they tend to mess up when structures are proteins that are in a complex or they're groups. So if you have a multimer, if you've got interactions with something else, those tend to be harder. And why do you think they would be hard pressed to get structures right when, say, it's bound to another protein?
There's not as much existing data on those protein-protein interactions.
Good. So what kind of data are they using as their input?
Essentially primary structure for a single protein, I'm assuming.
Yeah, they're using the multiple sequence alignment for protein A. And if protein A binds to protein B, a lot of the information about the distances is really from the correlated mutations in A and B, and they don't have B here. So you can imagine it's not going to be too hard if you know what the complex is that you're solving for, to take the next step and just try to make a potential energy function for that complex together, right? And so presumably some of these things are pretty easy to adapt the algorithm to overcome. There may be other things that prove to be a bit more challenging. Any thoughts on what kinds of conformational changes might be harder to predict with this approach?
Would something like a membrane-bound protein be very difficult to solve like this?
That's an interesting point. We'll come back to that. It's certainly reasonable to be worried about those. Anything else people think about?
I would think, like, just in general, protein complexes that have significant conformational changes, like depending on what function they're carrying out, like maybe motor or transport proteins, like I would think that would be harder because it's like over time changing and not one stationary shape.
That's really great. So if we think about motor proteins, right? So they have metastable states, right? They've got more than one strong energetic minimum that they can reside in. And I think we talked at one point about the viral fusion peptides, proteins that can adopt more than one really stable state. And so if a protein has multiple stable conformations that are induced by something else, so if it has two, let's just think this through. Let's say a protein naturally, without interacting with anything else, has more than one stable state. Should the AlphaFold be able to correctly predict those 2 or 3 states? Anyone vote yes or no? How many yeses?
Emily, of structures that they produce?
Sorry, I couldn't quite hear you. Say that again, please.
Like, would they show up in the family of structures? So like one of those 2000, and then maybe not. Obviously, if you only pick one optimal, then it wouldn't be in that singular one, but it would be within the family as a whole.
Exactly. So if the protein has multiple stable structures on its own, presumably that information is already embedded in the multiple sequence alignment. And so in principle, then in the same way that, with the true energetic, physical energetic minimum, you can reach the two energetic minima, presumably through the gradient descent, depending on what starting conformation you start with, you'll either descend towards one of those minima or descend to the other. Does that make sense? Any questions about that? So then the challenge becomes when there's some exogenous factor that changes it, right? And induces those conformational changes. So it could be interaction with another protein. Maybe it's an allosteric regulator that changes the structure of an enzyme. Think about hemoglobin having multiple states. Not all that information is necessarily embedded in the multiple sequence alignment. Depending on exactly what the details are, it may be that certain states are really, really hard to predict because we're missing information about what that other piece is that's coming in and causing the conformational change. So there may be some things for which it's really easy to extend to, and other things for which it's much harder to extend to. Okay, so in the science piece, there were some other good quotes as well. And this gets back to the question about the membrane proteins. So they said that the organizers were worried that perhaps they were somehow cheating because the results just seemed too good to believe, literally too good to believe. And so they actually gave them a special challenge. One of the contributors of experimental data had a protein structure that they've been really struggling to work on for a long time. He said for a decade they've been working on it. There weren't a lot of details here, but I imagine this is a protein for which they had diffraction data, but they didn't have enough information to solve the phase problem. So they weren't able to determine the structure from the diffraction data. So they passed this along to AlphaFold, and it's a membrane protein that this group hasn't been able to come up with a solution for a decade. And they come back with a structure, and they're able to stick that into the X-ray diffraction data, presumably by molecular replacement. And sure enough, it's completely consistent with the experimental data. There's no way to cheat on this, right? So there could not have possibly cheated this. I don't know how they do it. This is really, really astoundingly good results. And this is perhaps the best confirmation you get because we intuitively thought that membrane proteins would be really hard. Why? Because membrane has a role that we don't fully understand. There are not enough solved structures out there, perhaps. None of that seemed to be a barrier. Okay, and then the science article mentions some other things that they struggle with. So there are ones where there are these repeating units, and Jumper, who's the lead author on the abstract, says, well, they're working on it, and they have some ideas of how to fix that as well. So a couple of other things, quotes that really stood out in this. The organizer of CASP says, I had always hoped I would live to see this day, but it wasn't obvious that I was going to make it. Right? And when we look back at those plots of GDT versus difficulty, you can see why, right? First decade looked really great. Then it seems to be such a slow process, minor improvements, some new ideas of how to manage the contacts, but it really didn't look like there was going to be a breakthrough. And here it's just truly amazing. Okay, so when are we going to find out exactly how they did it? They plan to publish the work, but it's unlikely to happen until sometime next year. And they're exploring how to make this available. So a lot of unopened questions. I did mine their abstract for whatever little I can tell you about what they did. So these are some quotes. This is a quote from Nature that says that whatever AlphaFold 2 is doing, it's not based on what they told us 2 years ago, or I guess it was January of this year. They did try improving on the original AlphaFold. They hit a wall. And so here's what they do tell us. Something about additional information on the physical and geometric constraints. And they're not just trying to predict relationships between amino acids. That was that probability distribution over contact distances. But they simply go ahead and try to predict the final structure. And so this is not particularly helpful information. It's more complex. Well, I think we could figure that out. They do have this picture, and I guess you can try to guess at what some of the things are being done here. There is a point at which they're trying to predict pairwise distances, but some of them is going straight into predicting structure. There is this short abstract, and I'll post these notes later if you want to follow up on some of these references. What I'd caution, though, is what I saw with what they published 2 years ago, I should say their success 2 years ago, is that if you just read the abstract and then there was a quick paper that came out after that, it's pretty hard to make those two match up. And then it had very little relationship to what they published earlier this year. So maybe a while before we get a clear description of what went on. And this is not because of bad science or anything. It's just that the contests, people are constantly adapting during the course of the contest to seeing what they're doing well on, what they're doing poorly on, new ideas. And so describing the history of what you do over the course of the contest is different from describing the best ideas you have when the contest is over. But this is what the abstract says. Take it for what it is. The input is a sequence, the multiple sequence alignment, and a template. This sounds just like what we've seen up until now, right? That's just like CASP 13. And then they're using a deep learning method. And this is not particularly helpful, but it produces a variety of predictions. Okay, so they still predict distances and torsions. That's what we saw last time. But now they're also predicting atomic coordinates. And that's really new, right? The last one produced that potential energy function, and then they did gradient descent over the potential energy function. Now they're just directly predicting the atomic coordinates. And then this estimate of the C alpha IDDT, this is a measure of how close you are to your template. So clearly something has changed dramatically, and they're just jumping ahead, not having to do the gradient descent and just predicting the structure straight off. What helped the clue here is this attention-based deep learning. So attention-based neural networks are, somewhat recent advance in neural networks, where instead of trying to have the neural network learn everything about the problem, it tries to focus in on certain aspects of the problem that seem to be, according to the algorithm, the most relevant. And that may be the big difference. Again, we'll have to wait and see. And so it looks over the full multiple sequence alignment, but finds different pieces of the multiple sequence alignment, different regions that's going to give more literally attention to than others. And then I guess the last piece is that every prediction is relaxed using gradient descent with a force field. And this amber is one of the physics-based potential energy functions rather than a statistical one. So they get awfully close with the neural net, and then they tweak it at the very end using physics. And elsewhere it said that this last phase didn't dramatically change things. So, which is probably what you'd expect, right? Because it would take quite a long time to do a molecular dynamics simulation that would actually get you to the right structure. All right, any questions on what we've discussed so far? I'd like to spend a little bit of time telling you how I think this is going to change things in the field, and then I'd love to hear your thoughts as well. One big question is how accessible the solution becomes, and we don't obviously know that. Is Google gonna, be a good sport about this and make all the code available? And even if they do, is the architecture something that's really easy to implement? I think from some of the articles I've seen, it does seem that they didn't use some incredibly large compute cluster. It seems something manageable. So if the code becomes available, the hardware is not too formidable, then you could imagine that, you could start to apply this to every sequence alignment out there. Right? And so in the same way that we currently go to the PDB to get a known protein structure, you can imagine that in the not too distant future, there'll be a parallel one, which is purely predictions. And if you're trying to solve some molecular engineering problem in your lab, you go to the real PDB first. And if there's no structure there, go to the predicted PDB, and you probably have as good a chance of it being right as if it was, in the true PDB. We'll probably end up with some metrics that'll help us get a better sense of when the algorithm is likely to be wrong. But that said, there'll be some sort of quality score, and you can make your own decision as to whether that predicted PDB structure is going to be good enough. So what are the settings where you think where we already know it's not going to be good enough? What do we know that AlphaFold 2 is going to struggle with?
They mentioned the multiple protein interactions, so like multiple subunit proteins, for example.
Multi-subunit proteins, if you wanted to predict, say, a transcription factor binding to DNA, let's say you wanted to predict how a drug binds to a protein and changes its conformation. So there it's not going to be sufficient, but how can you leverage the predicted protein structure in cases like that? So let's say you're taking this course in 5 years, or you're teaching this course in 5 years is more likely, and you want to give the students assignment, and there's the predicted PDB, and I've got protein A and protein B, and I know they interact, and I only have the structure in the database of the prediction of free A and free B. What might you have your students do in the future?
Could you essentially use those two independent predicted structures as a starting point, and then you could use a physics-based or statistical-based model to kind of do the docking?
Absolutely right. So this is just going to completely revolutionize where we start. Right now, that problem is unsolvable. We have two proteins, neither of which we know the structure of. We want to get the complex. You don't even know where to begin. But if you've got the solved structures, now it depends obviously on how big a conformational change and whether it's going to be accessible to whatever refinement procedure, search procedures we're using. But having those initial structures is a huge step forward. And then in structure determination itself, I believe you talked about with Alan the phase problem in X-ray crystallography. Is that right? Not if that's true. Sounds familiar. Right. But in solving X-ray crystal structures, we need some way of resolving an ambiguity in the diffraction patterns. We need to get the phase. It's very hard to do that sometimes experimentally. And so in cases where you do know a rough protein structure, you can plug that rough protein structure into the diffraction data with something called molecular replacement. And that's what we saw in this example when they thought Google was cheating. They gave them this unknown protein structure, and then the people of the diffraction data were able to plug in the predicted structure and see whether it was right. And so I think that's going to be another boost. So let's say I do need some experimental structure. It's still going to be a lot easier to solve those things, because I have to grow one crystal into one condition, and then the molecular replacement becomes incredibly fast. So that's going to dramatically things. And then more experimental effort can go into solving the kinds of problems that that might be difficult to do with these neural net approaches, because there just isn't enough training data, whatever that may be, large complexes, or I was going to say membrane proteins, but apparently they're doing fine with membrane proteins, interactions with drugs. And so I would think it's going to dramatically accelerate the predictions, because all of these prediction problems, you've solved the first phase where you needed a good, accurate structure. And that's even before we think about how we can take advantage of the algorithms to address some new problems. And one case where I think we often could use this kind of approach would be when we have genetic variants. So if I were to sequence, any two people in this classroom, well, not in a classroom, in this virtual classroom, we'd probably have, thousands of locations in our genomes where we differ. And many of those will be in protein coding regions. And when we're trying to analyze genomic variants in real settings, we do this a lot in our own lab in the context of neurodegenerative diseases like ALS, you're often faced with a tough problem where you find some variant and it's maybe modestly statistically associated with a disease population versus control. But it's very hard to tell whether that mutation is really a functionally significant variant or whether it's just chance. And trying to predict whether some variant affects a function is in general a really hard problem. But how could you use the Google approach to figure that out? So I give you the functional protein and then somebody's sequence that I suspect might be not functional, and what could you do?
I guess if you wanted like a very high throughput approach, you could just run AlphaFold 2 on it and then compute the distance between the AlphaFold 2 product and the known structure. And presumably greater distance is more functionally significant.
Exactly right. So, you know, I can take all those variants and I can see, do they fold to the same structure, right? And if it's a protein of unknown structure, so I can see, as you pointed out, whether they come up with similar structures. Maybe one of them just doesn't refine at all. Maybe the variant doesn't refine at all. That suggests it's unfolded. Or if it does refine, is it a different structure? Or is the mutation at a site where we can then try to infer what the functional significance is? So I think that is going to be a real game changer for a lot of applications in trying to figure out genetic risk for various diseases. And then where's the field going to go? Well, I think not too many people are going to be working on tweaking the prediction problem for individual proteins, although there probably will still be a lot of technical work that needs to be done to make this work for all proteins. We'll probably find that there's certain classes for which it's harder. But there's going to be a lot more work on then predicting interactions. So instead of, as I suggested at the top of this list, using experimental methods, you know, how can we advance these the algorithmic approaches so we don't have to solve more crystal structures, but we can actually predict how binding to another protein changes things, or how a drug changes things. Maybe in the last little bit here, I'll just open up for your thoughts about, you know, how you think AI is going to alter everything we've talked about in this class. Do you think that there's something special about this problem? That's why AI worked, and all the other kinds of things we talked about, metabolic modeling, you know, distribution of drugs throughout the body, those kinds of things are immune to AI, that they're too hard for AI? Or do you expect things to be quite broad, and these kinds of Google approaches to really have an impact across the spectrum? So what do folks think? Are we all going to be building neural nets for all of our problems in the future?
So I think it's pretty likely that people are going to try that at least and see how far reaching it can be. Because I mean, it does seem really powerful for this specific problem. So I do expect a lot of people to want to try and apply it more broadly. But whether it works or not, you know, to wait and see, I guess.
Yeah. Other thoughts?
I think one problem that could still exist is the need for a lot of data to train the neural network. So a lot of problems, it might not be possible to collect enough data points to actually have like an accurate training algorithm. So I think there's still a little bit, I mean, not every problem can still be solved with AI.
That's a really good point. Do people have thoughts about what of the problems we talked about in this course, or outside of it, if you have other ones, which ones do you think are closest to being amenable to these kinds of approaches?
I
feel
kind of,
like
oh, sorry, please go for it.
I was going to say, like, problems that have high throughput methods of collecting data in general, because then you can quickly create a data set to train your model on.
Dylan, did you want to say something?
I think with the pathway design, my guess is there will be like some graphical AI models in the future that do a good job suggesting what things to knock out if you want to upregulate or downregulate a certain other product.
Those are both really great points, right? So, you know, where there are high throughput approaches, where the data collection is cheap, then these methods are going to work well. And obviously, the neural net would not have worked if we didn't already have a huge database of sequences and a sizable database of structures. So, you know, high throughput screening has been done a lot in the pharmaceutical industry, much more so than academia. And already, I think we've seen some roles, we didn't talk about it in this class, but there have been some impressive leads in trying to predict drugs that'll work in particular settings. What about pathway design, Dylan, do you think makes it amenable, or anybody else who wants to jump in?
I think those are experiments you could run fairly quickly, and potentially even like in the future robotically. And I think that, you know, what we kind of do now with pathway design is like basic, like on-off logical guesswork. So my feeling is probably like with some data-driven methods, you could improve upon that.
That's a good point. And there'll be direct experimental data on the pathways. And then the other aspect of it, of course, is because sequencing is cheap, we have a lot of information on multiple organisms, right? And so in terms of what the network architecture is of a metabolic reaction, knowing something about what other organisms are out there could be very powerful. Other thoughts?
It seems like a missing link right now is like the relationship between structure and function. So I'm wondering like if in the future it could not only predict a protein's structure, but also potentially its function as well.
Yeah, I think that's a really, really good point.
I think something to add on to that, if we're able to predict function or interactions directly from a DNA sequence, then directed evolution experiments could be completely done in silico, and then those resulting mutations can be directly plugged into a sequence for an enzyme you're trying to evolve as well.
Yeah, I think that's a good point. And you can think about that as a feedback loop, right? Maybe at the early stages, you still need the directed evolution for a while, but you get better and better over time because you generate so much information. And maybe sequencing some of the suboptimal sequences helps you as well.
Right.
Yeah. Other thoughts or questions?
Could we maybe use this to like learn some of the more nuanced physical rules for like how a sequence determines structure? So I think one of the papers we read did that for like some very small, simple proteins, but maybe we could like now extend that.
That's a really interesting question, and it gets to the heart of how interpretable different algorithms are. And it sounds like one of the big shifts between CASP13 and CASP14 is that they pretty much abandoned trying to do any approximation of a physical process. So that final neural network that they generate that just takes input as sequence and gives you coordinates, it may be very, very hard to back out of that any kind of information about why. And this is the challenge that we face as a field, where often it isn't too hard to get a machine learning algorithm to make a prediction for us, but we don't understand often why it is. There's a lot of interesting work right now on trying to make more interpretable AI models. And in many cases, you can distill down the insights from an uninterpretable model to make one that is interpretable. So that would require probably a lot of work. It won't be a trivial task. It's conceivable that that information is in there. Well, it must be in there, but it's conceivable that it's be extracted from there. But I think that may be one of the hardest things to do with this. It'll be probably much easier to take kind of rough insights about how AI was applied in this setting and apply the AI to another setting.
So I was thinking about maybe we could use AI to solve the 3D structure of genome, like to use the sequence and some other, I think, contextual information to predict whether, say, an area of the genome is, is more densely packed, or if it's accessible for transcription, things like that.
Yeah, I think that's a really interesting application. And one of the things that's always been rather mysterious is that you have transcription factors who have a huge number of binding sites throughout the genome, and they bind to a tiny fraction of all those binding sites. We normally kind of wave our hands and say it has something to do with chromatin accessibility and use rough measures of chromatin accessibility for that, but we can't really predict it. Maybe if you had a good three-dimensional model of the genome, you'd be able to predict that more accurately, predict which regions are being activated by which, and that could have a tremendous impact on our ability to model biological systems.
I was curious how you could incorporate post-translational modifications into this type of prediction software. Do you know about that?
In terms of what they've actually done, I know only what I've told you, what I read in the
Yeah.
New York Times, right? There's really very little information out there. What do you think? How do you think that's going to play out? What do you need to be able to solve a problem like that?
So I guess you couldn't do it from the DNA sequence, but if you could get a close approximation and then use like the more traditional modeling softwares, then maybe you could add it in post, and hopefully it's not changing the conformation too much. I don't know. I don't know a ton about like oscillation and all that.
Other thoughts on that topic?
I wonder if you could use the structure output of a model like this as an input to some other model that would perform those post-translational modifications.
Yeah, and it could be, I think it was Owen was suggesting that it's maybe a physical model or it could be a statistical model, right? And so maybe we can learn something about these changes. The other aspect is training data. So if you solve protein structures that have a lot of post-translational modifications, then you'll have the training data to solve that. And that may be where more effort goes. So instead of working on the protein as it gets produced in some non-native host, like you produce your protein in E. coli or whatever it is, and it's not modified, maybe that problem is more or less solved by an AlphaFold approach. But now we look at that protein in some other context where it has, we talked about complexes, but I think this issue of post-translation modification is really key. And we know lots and lots of proteins that are regulated primarily by post-translational modifications. So that could be a really big area of effort in the future. Well, I hope you found this this new topic interesting. Keep your eyes open. We'll, as I said, be learning a lot more. Probably take about a year before we really understand what was done there, and a lot longer before we understand the impact. But it is a truly remarkable accomplishment and worthy of quite a lot of praise. Nice talking with all of you. Take care. Have a great day.
Thank you.